With more than 6,000 small and micro-cap companies listed, if you're looking for the next apple at the earliest stage, then channelchek truly is the orchard. The listed companies support channelchek, so it's free for you, the potential investor, to gain access to institutional quality research from FINRA licensed analysts, advanced market data, industry reports, news, and a growing catalog of videos and webcasts. Make sure to subscribe to our YouTube channel and register for channelchek so you're always up to date on what's going on at the small and microcap data place. Welcome to the Virtual Roadshow series presented by channelchek. This roadshow features a formal corporate presentation from Rodney Varner, CEO of Genprex, NASDAQ ticker symbol GNPX. Research coverage of Genprex is provided by Noble Capital Markets, a FINRA-licensed, SEC-registered broker-dealer. You can find Noble's research on Genprex on channelchek.com or by clicking the link in the description. With that, I am pleased to present Rodney Varner.
Well, good afternoon, everyone. I'm Rodney Varner, one of the founders and CEO of Genprex. Thank you all for attending this presentation. We appreciate your time and your interest in Genprex. At Genprex, we're reprogramming the course of cancer and diabetes using gene therapy. I commend you to this forward-looking statement slide. I know you see it in every presentation, but it is important. Things may not go as projected or forecast. And by the way, you can see this slide and many others, a longer, more detailed slide deck on our webpage at genprex.com. This is our basic stock information you can see on your trading platform. Symbol is GNPX, trading on NASDAQ. I would point out that importantly, as of the end of the first quarter, we had 47 million cash on hand and no debt other than trade payables. At Genprex, we have a gene therapy platform for oncology. Now, let me point out that we have two separate and distinct programs. We have an oncology program that we've been working on for a decade based on technology invented at MD Anderson Cancer Center. And we have a diabetes gene therapy program that's based on technology invented and being developed at the University of Pittsburgh. I'm going to spend most of my time on our oncology program because it's much farther along and has had much more development. For our oncology program, we have a gene therapy platform that uses non-viral DOTAP cholesterol molecules to deliver therapeutic genes to tumors. This delivery platform is extremely important. Most gene therapies use a viral delivery system, which uses hollowed out viruses to deliver therapeutic genes. And in the case of cancer, they deliver them into tumors. And with most of those viral delivery systems, the virus has to be injected directly into the tumor with a needle, which means that the tumor has to be identified and it has to be reachable. With our platform, our gene therapy is injected by IV. It circulates throughout the bloodstream and attaches to tumors wherever they're found, whether they've been identified or not, and they don't have to be reachable with an instrument. Our lead investigator or inventor, Dr. Jack Roth, was one of the early movers in gene therapy. He led some of the earliest gene therapy trials in the United States. And early on, he used viral delivery systems as well. And he recognized the limitations of those and wanted to get away from them. And so he developed this non-viral system that, again, is systemic. And it's also shown to be a very safe delivery system with minimal side effects. We believe this is the first systemic gene therapy used for cancer in humans. And the words systemic and for cancer are key there. Again, we're not aware of anyone else using a systemic gene therapy for cancer. And we've already treated over 50 patients in clinical trials with this system. So it's not just theoretical. Again, we're not aware of anyone else who has treated even one patient with a non-viral systemic gene therapy in cancer. We're addressing large markets and unmet needs, and I mean really large markets and large unmet needs. Lung cancer kills more people than any other form of cancer, and it's the second most common form of cancer. And the market for lung cancer drugs exceeds $20 billion a year. We're combining our gene therapy, REQORSA, with top-selling cancer drugs. Tagrisso by AstraZeneca with sales of over $4 billion last year, and Keytruda by Merck with sales of over $14 billion last year. We have an FDA Fast Track designation for that combination with Tagrisso. We have demonstrated clinical achievement. We've treated over 50 patients in 2 clinical trials with our gene therapy. So we know a lot about it, and it's shown to have a very favorable safety profile with strong indications of efficacy. We're initiating 2 clinical trials in lung cancer this year, the one with Tagrisso and the one with Keytruda. We have world-class academic partners in MD Anderson in our oncology program and the University of Pittsburgh in our diabetes program. We have an expanding pipeline, as evidenced by our acquisition of the diabetes technology last year. We're working on additional potential technologies, both internally and for potential in-license, and I anticipate that our pipeline will continue to expand. And we have a strong balance sheet. As I showed you before, we had about 47 million as of the end of March, and no debt other than normal payables. So we're well financed and in a good position to continue to execute on our strategies. Here is our pipeline. The two trials at the top, Acclaim 1 and Acclaim 2, are the 2 trials that I mentioned, the one with Tagrisso, one with Keytruda. These are branded. The Acclaim 1 and Acclaim 2 are official brands of these clinical trials. As our drug is branded REQORSA, in order to market a drug, a company must obtain approval of the trade name from the FDA. We have done that, and we have the approval for the name REQORSA. It's a conditional approval in that they review it again before you actually begin to sell the drug, and it has to be cleared again, but it's the normal process that a company gets a conditional approval first, which we have done for the brand name REQORSA. After the Acclaim 2, you see below that ONC001. That was a clinical trial we did with REQORSA as a monotherapy, that is, using it alone in lung cancer. And below that, you see ONC002. This was a phase 1/2 clinical trial that we did combining REQORSA with Tarceva, which is an earlier generation of EGFR TKI inhibitors, and Tagrisso, which we're doing our current clinical trial in, is a later version of that type of drug, just a later generation. It was based on this earlier clinical trial with Tarceva, along with extensive preclinical data, that we were able to get the FDA Fast Track designation for use in combination with Tagrisso. And then we have a number of discovery programs that we haven't announced yet. Just want to mention that we're evaluating technologies both for in-license and also for development internally. And then finally, there's our diabetes program, which is preclinical. It's ongoing at the University of Pittsburgh. The university has previously published studies in mice on this therapy, and they're in the process of doing research in primates on this technology. So while it's early, preclinical, we're very excited about it because of the potential to treat both type 1 and type 2 diabetes. Again, just to delineate, we have two distinct programs. They're different gene therapies. One for oncology using our proprietary non-viral delivery system, and one for diabetes, which actually uses a more traditional viral delivery system and is being developed at a separate university, University of Pittsburgh. Now, more on the non-small cell lung cancer market. There are about 2 million cases of lung cancer a year worldwide, about 235,000 in the United States. About 84% of those are non-small cell lung cancer, which we're addressing. So you can see we're addressing a large majority of the market. Again, we're combining and have the fast track for combination with Tagrisso, 4 billion plus of 2020 gross revenue, and also with Keytruda, which had 14 billion plus of 2020 gross revenue. And the total market is expected to grow to 26 billion or more by 2023. So very large markets. And let me point out that we're not attempting to take market share from anyone. We're attempting to be combined with the two largest selling drugs in lung cancer and share in their markets and actually improve upon their revenues. So we don't have to fight giant pharmaceutical companies for market share. We have this novel platform. This further illustrates the platform. I explained it a bit earlier, but we have multiple therapeutic genes. We have a series of genes under license that could be used in cancer. We have, our researchers have proven synergies with a number of different lung cancer drugs. I've mentioned the two large sellers before, but there are a range of other cancer drugs that our researchers have demonstrated synergy with. So we can be combined with a number of different drugs with the potential to improve their benefit. And then we can also address multiple types of cancer. There's research from MD Anderson and other academic institutions that indicates that the therapeutic gene in our lead program called TUSC2 is implicated in many types of cancer and could potentially be therapeutic in those cancers as well. So we truly do have a platform with many shots on goal here. Let me expand upon our delivery system. As you can see in these diagrams, our nanoparticles are basically fatty molecules. They're DOTAP cholesterol molecules. They're engineered as to size and also with a positive electrical charge to be attracted to cancer. These nanoparticles encapsulate our therapeutic gene DNA plasmid and are then put into a liquid solution where they're injected by IV and they circulate throughout the body and attach to tumors. Biopsies of human tumor tissue has shown an uptake of up to 33-fold of these nanoparticles in tumor cells versus normal cells. So up to 33 times, or these cells, these nanoparticles go into tumors up to 33 times more than they do into normal cells. And even in normal cells, they don't have any activity because their function is to suppress tumors. And obviously, if there's no tumor, there's nothing to be done. So our researchers have found little or no toxicity in normal tissue from these nanoparticles. we've treated over 50 patients with it and shown a very favorable safety profile over all of those patients. There are three basic mechanisms of action that have been identified for REQORSA against cancer. There are actually probably others that haven't been as well identified, but it's known to control cell signaling. It's the method by which genes instruct cells on how to grow, how large to grow and how long to live. If those go awry, then tumors can develop. Similarly, REQORSA stimulates apoptotic pathways, which is programmed cell death. Our cells are programmed on how long to live, and if they don't die when they should, then that can lead to tumors. And our drug REQORSA stimulates programmed or normal cell death. And finally, and very importantly, it modulates the immune response. It promotes immune activity against cancer. This is why we call it an immunogene therapy. It has strong immunotherapy effects. These are simply further slides showing the modulation of the immune system by REQORSA, and you'll hear a repeated theme through the rest of the program that this synergy is not only for the combination of REQORSA with Keytruda, which is our initial combination drug in immunotherapy, but also Keytruda plus chemotherapy. And this is important because many of the patients who receive an immunotherapy like Keytruda receive it in combination with chemotherapy. So by showing synergy with both Keytruda alone and with Keytruda plus a chemotherapy, we potentially could treat a large majority of the patients who receive Keytruda, which is that very large market. This is data from our REQORSA monotherapy clinical trial. This clinical trial was done, this was our first clinical trial. It was done in late-stage lung cancer patients using REQORSA alone as a monotherapy. 23 patients were evaluated, and 5 of those 23 showed stable disease or better. 3 of them had tumor responses, which is shrinkage of their disease. This trial also showed that the drug was well tolerated in these patients. Now, this slide on the right, this is quite interesting. This was one of the patients in the phase 1 trial. This patient, as you can see by the arrows, had multiple tumors in her lung and pancreas. And after treatment with REQORSA, those tumors disappeared by PET scan. And this patient went on to live over 7 years after being treated. This was a stage 4 lung cancer patient with a very short life expectancy when she entered our trial. So this was a very gratifying outcome. Granted, this is an outlier. We don't expect this with a lot of patients, but it's very encouraging nevertheless. I got to meet this patient. We don't normally know who the patients are because of HIPAA rules. But MD Anderson published an article on this patient, and therefore we identified her and contacted her, and she agreed to meet with myself and other officers of our company, and we had a lengthy meeting, and she attributed her lengthy survival with after late-stage lung cancer to our gene therapy, which was really gratifying. This is data from the phase 2 portion of our phase 1/2 trial combining REQORSA with Tarceva. Tarceva is an earlier generation EGFR inhibitor, similar to Tagrisso. And in this trial, we had 9 patients that were subject to evaluation. And on the far right, you can see the various lines of therapy that those patients had prior to treatment with REQORSA. They all had multiple lines of therapy. Some of them had chemo, some of them had chemo plus immunotherapies, some had Tarceva, so they were all heavily pretreated, and 7 of the 9 saw a benefit after going on to our combination treatment, even after all of these prior pretreatments. And really interestingly, we see patients here who benefited after immunotherapy and after Tarceva, and we see patients who benefited who were EGFR negative, who you wouldn't expect to benefit from Tarceva at all, indicating that our drug is definitely making a contribution. One of these patients, the top one there, had a complete response, complete disappearance of their tumors. So we found this data very encouraging, and it was this data plus extensive preclinical data that led to our Fast Track designation that we're operating under now. These are important findings from our academic researchers. REQORSA plus Tagrisso reduced tumor growth in Tagrisso-resistant tumors. A major problem with Tagrisso and other drugs like it is that patients go on to them. If the patient is eligible, they have an EGFR mutation, they're called EGFR positive, they go on to a drug like Tagrisso and they receive a good benefit. Tagrisso is very good in the early stages after a patient starts taking it. But unfortunately, almost inevitably, the patients become resistant to it and no longer benefit. And so the challenge with these drugs is to overcome resistance so that the patients benefit longer. And our academic researchers have shown that REQORSA plus Tagrisso overcomes resistance in tumors to Tagrisso. So this is one of the foundations for our currently planned trial of REQORSA plus Tagrisso in patients who have already progressed on Tagrisso. This is the basic study design of our Acclaim 1 trial, which is the trial with Tagrisso. It's a phase 1-2 trial. There will be a dose escalation portion of it, about between 9 and 18 patients in that, and then a phase 2 with about 74 patients. An interim analysis is planned at about 51 events, which is disease progression or death. And the study arm is REQORSA plus Tagrisso. The control arm is continuing Tagrisso monotherapy after the first progression on Tagrisso. So it's the time between first and second progression. And other researchers have shown that time to be expected to be between 4 and 5 months. So we would hope to materially beat that time to the second progression. Again, in the phase 1 portion, the primary endpoint is dose-limiting toxicity. In this phase 2 portion, the primary endpoint is progression-free survival, with secondary endpoints of overall response rate, overall survival, tolerability, and number of adverse events. Now, as I mentioned, researchers have shown that REQORSA is synergistic with immunotherapies. And again, it's not just Keytruda, but other types of immunotherapy as well. This was an early study showing this synergy. This was with syngeneic mice, which I, as a non-scientist, kind of think of as normal mice. This study showed a strong synergy with the drugs receiving the combination treatment living or having substantially better survival than those that didn't. After that study, the researchers went on to do a study in humanized mice. Humanized mice are mice in which the scientists insert human immune cells and allow human immune cells to grow within the mouse. They can then challenge these human immune cells with cancer and treat the mouse and see what the response is, so that they can see the response in human immune cells. And they got similar results with these humanized mice. Again, there was synergy with REQORSA plus Keytruda, and also with REQORSA plus Keytruda plus chemo. Recently, in the studies published last month at AACR, the researchers demonstrated a rationale for first-line treatment of lung cancer patients with the combination of REQORSA plus Keytruda. They also identified mechanisms of action and also biomarkers which would indicate help indicate which patients would benefit from the treatment. So this was a very important study. It provides the rationale for first-line standard of care, which would be an additional clinical trial, but would open up, you know, a very broad market for REQORSA if it could be included in first-line standard of care. So this is very exciting, and we look forward to moving towards that goal. This is just the high-level overall design of the ACCLAIM 2 trial, which is the trial with Keytruda. It's planned to be a phase 1/2 trial of REQORSA combined with Keytruda in late-stage patients whose disease progressed after responding to Keytruda. It's planned to be REQORSA plus Keytruda versus chemotherapy in these late-stage patients. And we're looking forward to starting this trial soon. I have to point out the improvements that we made in manufacturing really over the past year. We really completed the technology transfer of our manufacturing from the academic setting, transferred it to commercial manufacturers, and scaled it up all last year. And we now have the ability to produce large-scale drug production through commercial manufacturers, which will support our clinical trials and ultimately commercialization. We're able now to produce the drugs in large quantity and store them in deep freeze for extended periods of time. This is critical to be able to ship to multiple clinical sites and, of course, in a commercial setting to commercial clinics around the world where the drug can be stored and then used as needed. Many gene therapies never bridge the gap from the academic setting to commercialized scaled-up production. So this is a really important accomplishment, and we think one to be proud of, and one that's really going to facilitate commercialization of REQORSA. A bit on our diabetes program. Again, this gene therapy is potentially applicable to both type 1 and type 2 diabetes. Those diseases affect about 34 million people in the United States, or about 10% of the population. This gene therapy has the potential, according to the researchers, to replace the daily burden of blood glucose monitoring and insulin replacement therapy in diabetic patients, which would be a huge thing. It has the potential to hold long-term effectiveness or even be a cure, according to those researchers. We just last month were awarded the License of the Year award by the University of Pittsburgh because of our work on this license in conjunction with the lead researcher, Dr. George Geddes. We were very happy to receive this and proud to receive it because the University of Pittsburgh is a very distinguished medical research institution with a long history and pedigree in medical research. It's the place where the polio vaccine was invented, for example. So we know there's a lot of great research going on there, many great licenses, and we received this License of the Year award. Also, last year, Dr. Geddes and his group received a $2.6 million NIH grant to support this research in primates. That's a large grant from the NIH, and those are heavily peer-reviewed. So I view that as validation of the technology by expert peer reviewers. And of course, it's great that Dr. Geddes and his group got that money to support the work in primates, which will fall under our license agreement of that patented technology. So very, very great and important developments on the diabetes therapy. We're looking forward to seeing more out of Dr. Geddes' lab. A little bit on how it works. It uses a novel infusion process using an endoscope and an AAV vector to deliver two genes, the PDX1 and MAFA gene, into the pancreas. These transform alpha cells in the pancreas into functional-like beta cells that can produce insulin but are distinct enough from beta cells to evade the body's immune system. This is important because other methods have been tried of restoring beta cells, such as beta cell transplants. And in type 1 diabetic patients, those new beta cells have been destroyed fairly quickly by the body's immune system, which is what happens in type 1 diabetes. So you could expect that with new beta cells. But these researchers have developed these beta-like cells that appear to be different enough from the originals that they're not destroyed by the immune system. And they're seeing a long-term benefit of these beta-like cells in the pancreas. Preclinical studies showed that this restored normal glucose levels in mice for an extended period of time. And a phase 1 clinical trial could be the first ever gene therapy tested in humans for diabetes. We're excited at that potential and believe that we have a good chance of accomplishing that. Here are some of our key milestones that we've recently completed, and then those that we anticipate over the next year or so. We completed the successful transfer of our manufacturing technology from the laboratory setting to the commercial setting. We've initiated clinical site recruitment for both our clinical trials. We've reported preclinical data for REQORSA in combination with both targeted therapies and immunotherapies. We've received centralized IRB approval for acclaim 1. An IRB is an institutional review board. It's a board that is charged with the obligation of assuring that clinical trials, really all clinical trials, are conducted. First of all, they're ethical and they're set up to be conducted in an ethical and safe manner. And one can't commence a clinical trial in the United States without having an IRB approval. So we're happy to have received that. We entered into an amendment of one of our license agreements to add additional oncology technologies and to expand our intellectual property portfolio. We're continuing to work on expansion of our intellectual property, and we have a really robust portfolio of intellectual property, which we are continuing to expand and bolster. Coming up, we believe that we'll receive FDA clearance to commence these 2 clinical trials. We look forward to dosing the first patient in each of them and announcing interim data from each of them, reporting preclinical data from our preclinical research under sponsored research agreements, both at MD Anderson and the University of Pittsburgh. We expect to explore new cancer indications, to continue to expand our pipeline and platform technologies, to pursue partnerships and collaborations, and to continue to expand our IP portfolio. People often ask me about comparable companies. We don't really identify comparable companies because we're not aware of anyone doing what we're doing. As I mentioned, we believe we're the first to have a systemic gene therapy for cancer in humans, and we're not aware of any other company using a nanoparticle delivery system for that purpose, and certainly one that has done clinical trials with it. But we put up here some companies that you might want to take a look at just and compare their valuations. And of course, there are many others that you might compare, but we believe that our valuation is very low compared to where it should be. And we believe that over the coming year, as we have achieved more milestones and garner more attention for our technologies, we believe we'll see that market cap rise. So again, we think we're really significantly undervalued, and this is a good time to get in. I'm going to skip over much of our management. There are a few people that I want to mention. First of all, our scientific advisory board. The chairman of our SAB, who's the lead inventor of our technologies, is Dr. Jack Roth from MD Anderson. He's internationally known and respected expert in both gene therapy and lung cancer. Dr. Tony Mok from the University of Hong Kong, also of that category, internationally known as an expert in lung cancer. Same with Pasi Jane and George Simon. Dr. Jane is at Harvard's Dana-Farber School. Dr. Simon is now at Moffitt Cancer Center in Florida. And then for our diabetes program, there's Dr. George Giddies from the University of Pittsburgh, who's the inventor of our technology. So a really accomplished scientific advisory board that we're very, very appreciative to have involved with our programs. And then we have a clinical advisory board also. I won't specify any individuals, but they're a very accomplished board of practicing clinicians in oncology. And then we have a regulatory advisor, Bill Gannon, William Gannon, MD there that you see. A very good clinical advisory board. And on our board team, I'll only mention one, and that's James Rothman, who is a co-founder of the company, has been a strategic advisor since the beginning. We founded the company in 2009, acquired our technology in 2010, and then in 2013 he won the Nobel Prize. And I like to point that out to demonstrate that we truly do have top-notch scientific expertise at Genprex. So we have 21st century gene therapies, large markets and unmet needs, combination trials with top-selling drugs, a Fast Track designation, and we're exploring new indications and new partnerships and collaborations. So with that, I hope you'll take a serious look at Genprex, and I thank you for your attention.
Thank you for joining us for this Virtual Roadshow presentation brought to you by ChannelchekK. View our YouTube channel for more video content, including C-suite interviews, virtual roadshows, and conference presentation replays. New content is added regularly, so subscribe below to stay up to date. Visit channelchek.com or click the link in the description to access equity research, news, and advanced market data on this and 6,000 other small and microcap companies.